Right, I hope everybody can see my screen okay. And thank you so much for having me. I'd like to thank the committee for inviting me to participate in this conference. I look forward to sharing some of the research my lab has been working on, as well as hearing some of the other talks from the other speakers on ways that we can optimize horse health during the various stages of horse health and development. So because we have such a broad audience today, which I think is fantastic, I'll start off my presentation with an introduction of what fetal programming is, how the field of research started, and then in order to understand what malnutrition is and what that means, we have to understand what the current nutrient requirements are, and then we'll go into the malnutrition piece. And we'll look at the effects on fetal growth, and then we'll go into effects on offspring metabolism and health that can persist into an animal's postnatal and performance career. And then finally, I'll spend a little bit of time on the effects of malnutrition on male offspring. This is a newer area of fetal programming, but we're starting to see some transgenerational effects associated with maternal malnutrition. And then we'll wrap up with some implications. We'll simplify things again, what it all means, and then provide some conclusions on how we can best manage these mares in an effort to promote fetal growth and development so that we can have healthy foals all the way through their performance careers. So when we talk about fetal programming, we're talking about any environment that's going to alter offspring phenotype. And this can come from malnutrition, which we'll spend the majority of our time on today, but will also can be coming from stress. So things like transport stress or stress coming from the housing environment or wherever that may be. It can also be associated with disease. So we know that there's a lot of physiological changes that occur during disease states. And so whether the disease actually functions to cross the placental barrier, or if the physiological changes that are occurring during that time can alter offspring phenotype as well. We also have things like air quality and extreme climate events that can be considered an alteration in maternal environment. And we don't have a lot of information on this from an equine perspective, but we certainly do more in the human health side of things. And then medications and drugs that may be administered during fetal development, critical periods of fetal development can certainly impact that, but we'll focus most of our efforts on nutrition today. So the field of fetal programming really started when following this, what we now know as the Dutch hunger winter between 1944 and 1945. And during this time, German occupied Netherlands faced a famine when food and fuel shipments were shut down caused by a railway strike. And that ultimately limited resources and resulted in rationing of supplies to a level that pregnant women during this winter were consuming somewhere around 400 to 800 calories per day. Now, 400 to 800 calories per day is about 6% reduction from requirements during gestation for women, where we recommend about 2,000 calories per day. So these effects were first published in 1976, where they found that children born to women pregnant during this winter had an increased incidence of type 2 diabetes, more cardiovascular health issues, and more high blood pressure. And all of these, what we know to be adult onset diseases, actually presented themselves much earlier in the lifetime of those people. So really significant effects here. And this is what really drove the field of fetal programming and ultimately led to a gentleman by the name of Dr. Barker developing his hypothesis known as the thrifty paradigm, where he went into the data and figured out that low birth weight causes an increased risk of disease. And then we've progressed in this field, and now we have what's called a match-mismatch theory. And what that means is the uterine environment is developed in such a way to prepare the developing fetus for the same lifestyle and same environment postnatally. So, for example, if nutrient restriction occurs during fetal development, then that uterine environment is preparing that fetus for a postnatal environment of nutrient restriction. Now, that's where the mismatch comes in, because typically we don't manage our foals on a nutrient restricted state. So if the mare is nutrient restricted during fetal development, that fetus is developing for a nutrient restricted environment. But when postnatally it's not nutrient restricted and oftentimes actually fed in excess, that's when we see an increased incidence of obesity and adult onset diseases that can persist throughout that animal's lifetime. So that's where we've progressed so far in understanding that these various factors that alter the maternal environment are ultimately going to alter the developing genotype of the fetus, what genes are turned on, what genes are turned off, and that influences both the placental and fetal phenotype, which is ultimately going to result in a change in that neonatal animal's phenotype. And that's going to cause various different changes in offspring phenotype that will persist through that horse's life. And that can be changes in metabolism, which we'll spend some time talking on a little bit later. It is also altering endocrine regulation, and that can be endocrine regulation of glucose homeostasis, of stress homeostasis. Cardiovascular health is a major one that we know of in human nutrition, but we haven't really dug deep into this on the equine side of things yet. And then interestingly, on the newer side, we're starting to see effects on reproductive performance that are ultimately transgenerational. So from a male side, we're seeing delayed testicular development, but we don't know yet how that's going to affect a stallion's performance later in life and whether that'll affect his subsequent offspring as well. So there could be effects of maternal malnutrition on, or maternal changes in environment, even down to her grandchildren and great-grandchildren. So it becomes very imperative that we manage these mares in such a way that we can support this fetal development. But what are the current nutrient requirements so that we can determine when we have a malnutrition event? So all of our nutrient requirements are ultimately based off of this fetal growth curve, where on the x-axis we have gestation in months, and the y-axis we have fetal weight. And I apologize for not converting this to kilograms, but the trend of the curve line will remain the same. So ultimately, we interpret this as the first 2 trimesters of gestation, there's not a whole lot of fetal growth, because what's predominantly occurring during this time is the development of organs and the development and maturation of the placenta. We need that placenta to develop in such a way during the first 2 trimesters of gestation that we can push a lot of nutrients to that developing fetus and support this time period of more rapid growth during 3rd trimester gestation. During this 3rd trimester, that's where we're seeing maturation of organs and organs starting to come online and be active. So for example, the fetal pancreas becomes active around the 3rd trimester, end of 2nd trimester mark, and starts regulating the foal's own glucose levels during that time. So it's really important to recognize that this period of rapid fetal growth is also a critical period in organ maturation and function as well. Interestingly, in mares, we see this anomaly during 3rd trimester gestation, where she becomes much more insulin resistant. So insulin resistance, just a simplified explanation of it, is where the tissues, so skeletal muscle and liver, no longer respond to insulin that's released by the pancreas to control glucose levels. So you can think of it as kind of like the annoying little brother. Eventually, you're just going to stop paying attention to him. That's what insulin does. So insulin will continue to be released, but ultimately the skeletal muscle and liver do not listen to that insulin anymore. So if the mare then doesn't take up her own blood glucose levels, those are then shunted to the foal in a prioritization to give those nutrients to that developing fetus. So based off of this growth curve right here, that's where all of our nutrient requirements have then come from. So during early gestation, we have really slow fetal growth. And so during those first 2 trimesters, we can really feed mares to meet their maintenance requirements. We don't have to increase anything really during those first 2 trimesters. However, once we get into those periods of more rapid growth during the 3rd trimester of gestation, we see increases in digestible energy requirements, crude protein requirements, as well as calcium and phosphorus, because ultimately we're trying to build a new skeleton for that developing fetus. And this magnitude of increase for these nutrients is about 20% above that of the horse's normal maintenance requirements, which is great for us that it doesn't go up any more than that, because as you can imagine, you have a developing fetus in a mare's abdomen, it's going to take a little bit of room and capacity away from her GI tract. So her voluntary intake actually goes down, which means we need a pretty high quality feed stuff during 3rd trimester gestation so that we can meet this increase in maintenance requirements. So understanding what those maintenance requirements are, now I can understand what malnutrition may be. And malnutrition can refer to overfeeding or obesity, which we know to occur in about 50% or more of our horse population. So that's where most of our data has come from so far. And that can be excess in the form of energy, which breaks down into carbohydrates and fat, or protein, or a combination of both. And in fact, most of the time it is a combination of overfeeding both, particularly when you're buying a commercial feed. Nutrient restriction, on the other hand, is underfeeding, where nutrients are restricted, not being fed at a level to meet nutrient requirements. And again, it can be energy and or protein, but usually a combination of the two. In our horse populations, this usually doesn't come from underfeeding from a volume perspective, but more from a reduction in feed quality. So if we have a bad hay year, or there's not enough pasture, or there's not good nutrient quality of that pasture, we can end up in a nutrient restricted state without even really realizing it sometimes. But the way we can assess mares from a nutritional status perspective is using what we call the body condition scoring system that was developed by Henneke and Associates in 1983. And that's depicted in this figure right here, where we ultimately assign a score of 1 to 9 for each of these locations on the horse's body, with 1 being completely emaciated, no fat cover at all, to 9 being morbidly obese, and 5 being right there in between. So I'll palpate this horse and I'll assign a score of 1 to 9 along his neck, along the withers, the ribs and behind the elbow, the crease down his top line, as well as on the tail head. And these are all locations where horses tend to deposit fat. And so I can assign a score to these to determine nutritional status. For our reproductive mares, we want them to be around a 6 compared to our performance horses, where we recommend them being at a 5. And that's a nice cutoff as well. So I can look at a horse, and if I can visually see their ribs while they're standing square, they're below a 5. If I cannot see their ribs while they're standing square, they're a 5 or greater. So kind of nice little cutoff to put that into a little bit of perspective there. But it becomes challenging for us to use body weight to monitor a mare's nutrition status, because we anticipate her body weight will go up during gestation simply based on her developing a foal and growing another body within her. So an increase in body weight during gestation isn't necessarily reflective of a change in nutritional status. So that's why this body condition scoring system was developed. So how does this relate to fetal programming? So I'll go into our current state of knowledge, and I'll start by explaining how we have researched this field thus far, and then what we need to go into next. So most of our research from a malnutrition perspective in fetal programming in horses has been done on overfeeding or obese models because of the prevalence of obesity in most of our horse populations. So this is much more well researched, and we've used 2 models so far to study this. Started out using what's pretty standard in the industry, where the control diet was a hay-only diet. The mares didn't receive any additional nutrition other than a high-quality hay source. And then the overfed group received both a source of hay as well as a source of grain to account for that. Now, what we found is that hay only is actually not sufficient to meet maternal requirements, and I'll show you how we figured that out. So we actually modified from there our system where we developed more targeted nutrition programs where our control diet was specifically formulated to meet 100% of nutrient requirements for mares during gestation. And our overfed group was modified to be exactly 40% above that of the control horses. And so that equated to mares receiving about 4 to 5 pounds per day, or about 2.5 to 3 or 2.5 kilograms or so per day of a commercial formulated mare and foal feed. So that's actually pretty standard practice for a lot of places that are feeding grain. Nutrient restriction, on the other hand, is not well studied in the horse, and we certainly do need to look into this more. But so far, we have had to extrapolate all of our information on nutrient restriction from other livestock species, predominantly cattle and sheep, and I'll present some of that information as well as we continue on. So this slide gets a little bit busy, but I'll walk you through it. The figures down here on the bottom relate to maternal body weight changes when we apply these treatments to them. So I mentioned in the previous slide that we started out fetal programming with a hay-only diet or a hay plus a concentrate feed. So hay being dictated by the dashed line, hay and grain dictated by the solid line, with day of third trimester gestation on the x-axis and body weight in kilograms on the y-axis. So we found here that mares during third trimester gestation that were on the hay-only diet did not increase body weight at all. They maintained their body weight throughout third trimester gestation. Now we know that mares need to increase body weight relative to fetal growth during third trimester gestation. So that told us that this diet probably was nutrient restricted, whereas our overfed diet was probably a true overfeeding diet. So we didn't really have a true control here. But interestingly, we didn't see an effect on the foal's birth weight after the time of birth. So then our next study, we modified our nutrition requirements. Same x-axis being gestation day. This is third trimester gestation. And this on the x-axis is change in body weight during that time in kilograms. So control horses fed exactly 100% of their nutrient requirements during third trimester gestation, gained around 26 kilograms during that time, which is exactly what we would expect for fetal growth. But our group that received 40% above that of their gestation nutrient requirements gained about 49 kilograms during third trimester gestation, letting us know that we did achieve that goal of a true control diet as well as an overfed group. But in both of these studies, regardless, we found that maternal body weight and body condition score was actually not strongly correlated with foal birth weight. And that's because a mare strongly prioritizes her nutritional her nutritional status, all of her stored nutrients towards the developing fetus in an effort to prevent that fetus from being in a nutrient restricted state. So in this study right here, these are our data from full body weight. So 41 and 42 kilograms in the control and high group respectively. Now, there are some exceptions to this on extreme, but all of this ultimately relates to nutrient transport and placental efficiency. So the placenta is tasked with moving nutrients to the developing fetus, so the fetus has nutrients available. So we've already talked a little bit about how mares lose a lot of their insulin sensitivity during third trimester gestation in an effort to shunt nutrients over to the developing fetus. And that's what we saw in these glucose tolerance tests that we performed on mares during third trimester gestation that were on those same 40% above control diets. So we found increased peak glucose concentrations as well as an increase in insulin released by mares on that overfed treatment group, indicating that she is working much harder to regulate her own blood glucose levels in an effort to shunt that glucose over to the foal. Another method we can use to determine nutritional status is by looking at a hormone called leptin, which is very strongly related to overnutrition or obesity. Oh, I apologize. So what we found is that mares that were overfed during third trimester gestation almost had a twofold increase in leptin concentrations, telling us that we did in fact have an overnutrition model, and that mare was producing quite a bit of leptin in response to that overnutrition state. Now, on the other hand, non-esterified fatty acids, also known as NEFAs, are an excellent indicator of nutrient restriction, because if a mare has to tap into her stored fat, she is going to release some of those free fatty acids into her circulation, and we can measure those. And so in this particular study, because it wasn't a nutrient restriction study, we saw no difference in those NEFA concentrations. But that also told us that our control diet was a true control diet, whereby we met nutrient requirements for those mares. But if this were a nutrient restricted study, I would expect the exact opposite results, where we wouldn't see a difference in leptin, but we would see a drastic increase in those non-esterified fatty acids in the group that was in a nutrient restricted state. So what does this all mean from a metabolism perspective? Most of what we have looked at so far is related to glucose homeostasis and the ability of foals to be able to develop all of the organs necessary to maintain glucose levels, to be able to use glucose efficiently as a source of energy, which is imperative for performance, and then be able to sustain that throughout life. So we start the story at the pancreas, because the pancreas is ultimately what the organ that's responsible for detecting blood glucose levels and then releasing insulin, in response to those glucose levels. So it's important to understand when development occurs. So the pancreas develops from an organogenesis perspective pretty early on in gestation, in the first 2 trimesters, but it starts regulating the foal's own glucose concentrations by 3rd trimester gestation. So important to have this develop appropriately. So we'll break down the anatomy and the molecular side of the pancreas. So the pancreas can be broken down into 2 different sections: the endocrine pancreas, which we'll spend the most time on, and the exocrine pancreas, which is responsible for releasing those digestive enzymes during postnatal life. But the endocrine pancreas is what we were most interested in this study. And the endocrine pancreas is made up of these islets of Langerhans, which are these little pockets of specific endocrine cells. And I have a nice picture depicting that in tissue in the next slide. And the cells that make up those islets include beta cells, which release insulin, alpha cells releasing glucagon, and delta cells that release somatostatin. And ultimately, these 3 hormones work together to control glucose homeostasis. Now, if we have a malnutrition event, the pancreas is very much so impacted by this. So in a nutrient restricted state, we see a reduction in beta cell mass. And that's going to lead to type 2 diabetes. So what ultimately happens, and I'll point you to this figure down here, is in a normal healthy animal, we have these islets of Langerhans dispersed throughout that have fully functional beta cells that release insulin in response to elevated blood glucose levels. But when we see impaired pancreatic function, it happens in progression. So first, we'll see what's called beta cell compensation, where we see an increase in functional beta cell mass and an increase in insulin release by those beta cells in response to hyperglycemia. And then that will eventually progress as those tissues continue to ignore that insulin, where we'll see decreased functional beta cell mass associated with beta cell failure. So in our nutrient restricted animals, we jump right to this beta cell failure. But in an overnutrition or obesity state, it depends on when that nutritional insult was applied. So if we apply during early gestation, we see this reduction in beta cell mass. But if we wait to increase that mare's nutrition, which we typically do wait to increase her nutrition till third trimester gestation, we actually see more evidence of that beta cell compensation with an increase in beta cell mass and number. And that's what we've seen here in this same study where we fed mares 40% above their nutrient requirements or control. These are stained slides of pancreas. And these unstained little circles in here are those islets of Langerhans. And so we took these slides and we determined how many islets were present. And islets are associated and very tightly correlated with beta cell mass and number as well. So we found almost a 6 or 80% or so increase in islet number and a twofold increase in pancreatic islet size in these foals from overfed mares. So we're basically putting these foals at a state of what would be similar to prediabetes in humans and getting much closer to that true insulin resistant state, even during that fetal development time, which can have significant implications as that horse progresses in life. But what does this mean for other tissues involved in glucose homeostasis? So we went to the skeletal muscle, because skeletal muscle is the most sensitive to insulin and plays the biggest role in responding to insulin to take up and store glucose as an energy source and maintain blood glucose levels. So similar here, skeletal muscle development and the effects of malnutrition are very much dependent on the timing of that nutritional insult. So should that happen during first trimester, we'll actually see these progenitor cells, which are basically immature cells that haven't differentiated yet, go more towards fat cells than muscle cells. But as we get later into gestation, we actually see more differences in the maturation of those tissues, so the maturation of fat cells, as well as the maturation and growth and hypertrophy of the skeletal muscle cells, which obviously we all know that we use horses as athletes. So we need those muscles not only to function properly, but also to have normal metabolic processes to be able to use energy substrates in a way that that horse can perform at the level we're asking them to. So we've seen quite a few different implications of maternal malnutrition on the skeletal muscle. We've seen shifts in muscle fiber type, which I'll go into a little bit more detail on. We've also seen changes in fiber size, as well as altered nutrient metabolism, meaning we may be affecting these horses' ability to utilize the energy available to them. So this figure right here came from the same study of 40% overnutrition. The blue icons right here, blue stained fibers are type 1 fibers. The green and the black fibers are the two different type 2 fibers, much larger in size. So what we actually saw here was a shift towards type 1 skeletal muscle fibers in horses that were overfed. We also saw an increase in muscle fiber size, which has been strongly correlated with altered nutrient metabolism and more impaired metabolism, and we'll dive deeper into that in a few minutes. So these are our data on muscle fiber size. We took 3 different muscles from these foals, a biopsy of those, the gluteus medius and the semitendinosus being 2 hind end muscles that play a really big role in propulsion and the motor of the horse, driving them from their hind end, and the triceps brachii muscle, which is a muscle of the forelimb that is a very stabilizing muscle. But in all 3 of these muscles, we saw an increase in fiber size in response to maternal overnutrition, which we think is going to contribute to impaired nutrient metabolism within those muscle cells. Oh, I apologize, I'm skipping around. So we also saw shifts in skeletal muscle fiber type. So a reminder for those of you and lesson for those of you that aren't familiar with fiber types, type 1 skeletal muscle fibers are slow twitch fibers. They're slow to contract and slow to fatigue. We see those predominantly in our endurance horses that work long and slow. Type 2 muscle fibers, on the other hand, are those that are really fast to contract, but they're also really fast to fatigue. Now, in our endurance horses, Arabians, those type 1 fibers are really important. But in our quarter horses that are meant to run the fastest quarter mile, we need the fast, powerful muscles functioning for us. So this shift from type 2 to type 1 skeletal muscle fibers that we saw in all of our skeletal muscles that we took may or may not be a good thing for the horses that we have working for us. This study was done in quarter horses. So this shift in favor of type 1 skeletal muscle fibers may not actually be a good thing. And we do know that type 1 skeletal muscle fibers, even during training, will not convert into type 2, and conversely, type 2 muscle fibers will not convert into type 1. So that horse is kind of stuck with those fibers as it moves into its performance career in early training. But what does this all mean from a metabolism perspective? And this figure here gets kind of cumbersome, but basically, to simplify it, muscle cells have this insulin receptor right here. So remember, insulin is secreted by the pancreas, binds to its insulin receptor, and then all of these various pathways are activated in the cell, and they tell this glucose transporter that just kind of hangs out waiting in the cell to move to the cell membrane and become active and bring glucose into that cell. So what we wanted to look at from a glucose metabolism perspective to pair with our pancreas data is the gene expression of this insulin receptor and this glucose transporter. But then also we wanted to look at some of the functional components within these downstream signaling molecules to see if they may be implicated in some of this work. And so we found that maternal obesity or overnutrition actually downregulated in the gluteus medius the glucose transporter. And remember, the gluteus medius is the #1 muscle that needs to produce all the power from the horse's hind end. So we may have a limited ability to uptake glucose into the gluteus medius to be able to use as a source of energy. And to support this, remember those downstream signaling molecules within the cell, we found an increase in some of those markers, which have been previously associated with insulin resistance. So not only do we have less expression of the glucose transporter gene, but we also have altered metabolism within that cell to tell that glucose transporter to take in glucose, meaning there's going to be less glucose available to the cell to use for energy, and they're not going to be able to use it quite as efficiently. So what are the long-term effects of this? Well, interestingly, when foals are born, they have normal insulin sensitivity at birth. But 2 studies, similar to the ones that we have performed, found reduced insulin sensitivity in these foals by 6 months of age. Now, insulin resistance and equine metabolic syndrome historically doesn't show up until horses are in more of their geriatric years. But we're programming, by overfeeding these mares, we're programming these foals to start showing signs of insulin resistance as early as 6 months of age. And these data probably start to explain at a molecular level why that's happening. Now we'll take a little bit of a turn because in this study, we found some things that we weren't quite expecting. So the spleen is a very important organ to exercising horses. It has a role in red blood cell storage. And when a horse is exercising at maximum capacity, the spleen will actually contract and dump its red blood cell contents into circulation to increase oxygen carrying to the tissues, to be able to prevent or delay the onset of fatigue during those intense exercise bouts. But it also plays a role in the immune system, both the innate and adaptive immune responses. And in this study, we knew beforehand that the spleen was incredibly sensitive to maternal nutrition, but we didn't know what that would truly mean. But we did find an increase in spleen weight by about 25%. So we don't really know much about this yet. We've only gone so far as to do a little bit of staining to see what's happening. So we still have a lot of work to do on this and are currently doing some of this in the lab right now. And what we found is the maternal overnutrition resulted in a 25% increase in the organ's weight and a shift from white pulp, which controls the immune portion of the spleen's function, towards red pulp, which controls the red blood cell storage. So what does that mean? Well, if there's more capacity for red blood cell storage, that could be good for us from a performance horse perspective. But if we're taking away that white pulp, then we may have some implications associated with immune health and immune function that may leave those animals more susceptible. But again, we don't know any of that yet because this is the only data that we have so far on this. So we're currently diving deeper into this to see what implications it may have. And then lastly, in our study, we found that the testes from foals of overfed mares were about 50% larger than that of foals from the control mares. So we wanted to look into this a little bit further, and this is a project that my graduate student Alex is currently working on in the lab, where we're looking at gene expression of particular genes that are associated with spermatogenesis and testicular function. We're looking at the histomorphology of those tissues to see if there in fact is more functional tissue in those testes, so more seminiferous tubules, meaning greater ability to produce more sperm. But we're also looking at some transcription and growth factors that are associated with maturation and development of those testes to puberty. So in this study, that's as far as we've gotten so far, but we dug into the literature and we found that there's actually another study that did the same thing in standard bred horses, overfeeding those mares during mid to late gestation, and they found significant delayed testicular development. And so we hypothesized we'd probably see the same thing in our study. But we don't know much about this in any of our livestock species. So there's a lot of work still to be done on this. So what does all of this data mean, and how do we simplify it in an effort to optimize maternal management, gestational management of our mares? So basically, we can take this all down to maternal malnutrition altering the offspring's phenotype in such a way that they're at greater risk of equine metabolic syndrome. They're at greater risk of obesity, which by default increases their risk of laminitis. And all of this can impact their performance longevity as well as their genetic performance potential. You know, we're pretty good at looking at genetic papers, registration papers, and so on to determine an animal's genetic performance potential. But if we didn't manage that mare in such a way to optimize fetal health, then there may be no capacity for that foal to actually meet what its genetics say it should be able to do from a performance perspective. And then again, we're learning what effects there is on that reproductive potential that may have transgenerational effects on future offspring. So essentially, we know from this data that maternal malnutrition can limit genetic performance potential by altering the metabolism of the muscle. The muscle ultimately needs to work for us from an athletic perspective. And it's going to change its ability to adapt to training. So these horses may have to take longer in those training years to be able to adapt to that, but they still may not be able to meet that genetic performance potential. And that in part could be because the shift in those skeletal muscle fibers. So how can we use this information to better manage mares? Well, we want to feed mares to meet requirements. We don't want to be feeding in excess. We don't want to be feeding in a restricted state either. So we like to manage our mares at a body condition score of 6 because we have plenty of data to suggest that when a mare is below a body condition score of 6, she has reduced pregnancy rates. It takes longer to breed her back, and it takes more breedings to get her to take. But greater than 6 results in a predisposition to adult onset diseases in her offspring. So we want to avoid any excess or restriction, and that is both volume as well as quality of those feedstuffs. And then we want to start increasing a mare's nutrition around that third trimester of gestation so we can support that period of rapid fetal growth. But we also have to recognize that her voluntary intake is going to decline during that time as well, because as you can imagine, a foal taking up all of this space within her abdomen is going to take away from the capacity of her GI tract. And then we have to prepare for the drastic increases in maternal nutrient requirements during lactation as well. So when we change a mare's diet to account for this increase in nutrient requirements, that again is about 20% above that of her maintenance requirements, we want to change and add that in slowly. And so good rule of thumb is for it to take about 7 to 10 days for us to increase her nutrients that she's receiving from feed. And this could be just increasing the volume of feed that she's getting, or if we're changing her feed completely, it should take about 7 to 10 days. And we want to start increasing this at or just before third trimester gestation, so we can account for those increases in energy, protein, calcium, and phosphorus demands. So this table here is just a nice protocol that can be used to change a mare's diet over a period of time. And this is about as fast as I will go in changing. But we want to make sure that when we change diet, we still have some of our old feed on hand. So on days 1 and 2, I'll feed 75% of her old feed, 25% of her new feed. Then moving to days 3 and 4, we'll go to 50% of each. Then day 5 and 6, we'll just invert that and start feeding her majority, 75% of her intake as new feed, 25% as her old feed. And then by day 7, we have her shifted over to her new diet that she can maintain through the rest of gestation. So to wrap up and develop some conclusions from all of this, maternal diet does influence offspring health and performance potential. And it does so from a metabolism perspective that we know at the pancreas and skeletal muscle level, there's still a lot to learn with other tissues. We know it can alter performance potential, and that's predominantly through skeletal muscle metabolism, but also what potential effects may the spleen have on this as well. And then reproductive potential from testicular development. We don't know much about female development and reproductive efficiency following maternal malnutrition. So this is certainly an area that we need to dive quite a bit deeper into. So we need to figure out what impacts there are on future reproduction performance, metabolism of these horses as they go into their performance and training careers, and what effects nutrient restriction has, because obesity does take priority in our industry, but we can't forego the fact that we have years of drought, we have years where hay quality is poor, and we may have greater prevalence of nutrient restriction in these horses that we don't even really know about. So before I completely wrap this up, I'd just like to acknowledge some of the funding agencies that have supported this work, as well as some of the folks that have been integral in getting this work done, and my 2 graduate students that are currently up in the lab working on getting us some more data in this field. And I know there will be time for questions after this session, and I'd be happy to answer any of those for you. Thank you again to the committee for inviting me to speak here, and I look forward to the rest of the conference.